We will.
For those of you who are just joining us, we will be starting shortly. We want to give everyone a couple of minutes to get in here, and we will get started at about 7:02. Thank you.
Can we see the participant list?
I don't think oh, there they are. Yep. So we're at 48, 49.
Yeah, it's going up.
Okay.
Connie is raising her hand. For those of you who are coming in or have questions, go to the chat and write it in. We have people that are backstage, so to speak, and we'll see your questions or your concerns. So feel free to go to the chat and punch in there any questions or anything you may have, because I see Connie has her hand raised. So Connie, I would go to the chat room and our colleagues will see your message in the chat room. Thank you. Well, might give it one more minute.
There we have 61.
Yes, we see. All right, just another couple of seconds. Numbers are going back up again. Well, I want to be cognizant of everyone's time, and so we are going to begin. Good evening, everyone. My name is Tom Cardia. I'm a Senior Vice President at the Diabetes Research Institute Foundation, but more than that, I'm a dad to 2 children who have lived with this disease for almost all of their life. I could not be happier that you are joining us tonight for the Diabetes Research Institute Foundation and the Diabetes Research Institute Foundation Canada, which you will hear the phrase DRIFCAN used, and that's what it stands for this very special occasion. We are so fortunate to hear directly from 2 prominent pioneers, and dare I say icons, who I've known for so long and have laid so much hope at their feet to help find a cure, and are well known in our diabetes research space. We have a worldwide audience tonight, from our friends in Canada to Italy to England and beyond, in addition to our friends across the United States. Our goal and hope for those of you tuning in tonight is that you're able to gain valuable insight into the type 1 cure-focused research, not through just one scientist, not through just one team, but through a journey of collaboration that is and has been happening between our 2 institutions and others around the world. In the interest of time, I want to give you just a quick snapshot of the 2 organizations that are represented tonight. The Diabetes Research Institute is a leader in cure-focused diabetes research. As one of the largest and most comprehensive research centers dedicated to curing diabetes, the DRI is aggressively working to develop a biological cure for diabetes by restoring natural insulin production and normalizing blood sugar levels without imposing other risks. The Diabetes Research Institute Foundation Canada, DRIFCAN, was established in 2005 to fund the Edmonton Protocol. Dr. Shapiro and his team are located at the Alberta Diabetes Institute at the University of Alberta in Edmonton, Alberta, Canada. We believe that donors supporting diabetes research want as much funding to go directly to cure-based research as possible. DRIFCAN funds clinical and human trials with Dr. Shapiro and his team. At this time, I'd like to introduce Melanie Hibbard, the Executive Director of DRIFCAN.
Thank you very much, Tom. And thank you everybody for joining us. This is definitely an exciting time. So as mentioned, I am the Executive Director for DRIFCAN. But more importantly, like Tom, I am a mother of 2 children who live with type 1 diabetes. So this is a very, very personal story and journey for me and why I'm involved. People often ask me why I'm involved. And obviously it's because of my children. But it's an easy one. I want a cure. Bottom line, that's what I want. And that's why we do what we do. I had heard Dr. Shapiro speak a few times, and I knew I had to do whatever I could to help him find the cure. This opportunity came my way to participate and be a part of the Diabetes Research Institute Foundation Canada, or DRIFCAN. And I can't say how many times I've gone, phew, that's a mouthful. And always wondered, oh, that's a lay name that I got to explain to everybody. So I just wanted to give you a little bit of background. And it actually leads back to Dr. Ricordi and Dr. Shapiro as to why we are DRIFCAN and why we're here. It goes back, DRIF was established first and was helping fund the amazing Dr. Ricordi and his research at the DRI Institute in Miami. Back at the time, it was obviously that we had to find a way to find a way to fund this cure research. And Dr. Shapiro needed the same support that Dr. Ricordi would need. So hence the starting of DRIFCAN, the Diabetes Research Institute Foundation Canada. But along with that, many people ask me all the time, are you associated with DRIF and the DRI in Miami? And the answer until now was no, we are not, but we are now. And this is an exciting time. DRIFCAN and our supporters here in Canada are very excited that for the first time ever, it's a partnership with foundations cross-border internationally. It's not been done before. And we know, and here's the thing about our foundations, DRIF and DRIFCAN, we hear our supporters, we hear our donors, we hear the people who want a cure. We know that you say that you want there to be a collaboration between researchers who are searching for the cure, but more importantly, we also want those organizations to work together. So we hear you, we're listening. And one day, like this, we're going to have a celebration of a cure. And these 2 doctors are going to be the ones leading the charge. So thank you again for participating and supporting us. We really appreciate it. And I look forward to this evening.
Wow, well said, Melanie. Very well said. Thank you. We're going to get started. I'm going to read a little bit about our 2 guests, but before we do, just a couple of housekeeping things. This evening, you're going to hear from both Dr. Ricordi and Dr. Shapiro on their thoughts about the state of type 1 research and the importance of collaboration through our virtual discussion. During the discussion, feel free to ask questions and our team will respond. If we're not able to get to your question during the allotted time, please contact a member of the DRIF or DRIFCAN team, and we will get back to you probably within the next week. This session will be recorded so everyone knows and will be released at a later date so you can share with your friends who could not join us tonight. I now have the absolute personal and distinct honor to introduce our 2 guests. Dr. James Shapiro was born in Leeds, England, and studied medicine in Newcastle and trained in surgery in Bristol. He developed a long-standing interest in eyelid transplantation as a medical student. He joined the faculty at the University of Alberta, U of A, in 1998, where he now holds the Canada Research Chair in Transplantation Surgery and Regenerative Medicine. He directs the living donor liver transplant and the eyelid transplant programs in Edmonton. He was the lead investigator on the famous Edmonton protocol, islet cell transplant treatment for diabetes. It had never been proven before that we could take islet cells from one person and put them in another. He is the Project 1 lead for the Canadian National Transplant Research Program, a national initiative designed to increase organ and tissue donation in Canada and enhance the lives of Canadians for whom transplant offers a second chance at health. Dr. Shapiro is actively researching ex vivo organ transplant repair. This year, his team began the first human trials with re-engineered stem cell-derived insulin-producing cells transplanted in an immunoisolation device. Dr. Shapiro is the recipient of the Hunterian, is that how you say it, Doctor?
You got it.
Hunterian Medal from the Royal College of Surgeons of England, the Gold Medal in Surgery, is a fellow of the Royal Society of Canada and a fellow of the Canadian Academy of Health Sciences. Again, pioneer, icon. Dr. Ricordi is a professor of surgery and director of the Diabetes Research Institute and Cell Transplant Center at the University of Miami. His research interests include diabetes cure-focused research, cell transplantation, and regenerative medicine. Dr. Ricordi is well known for inventing the technology that made it possible to isolate large numbers of pancreatic islets and for performing the first series of successful clinical islet transplants that reverse diabetes. The procedure is now used worldwide. For over a decade, he chaired the NIH Clinical Islet Transplantation Consortium that successfully completed the first FDA phase 3 trials of islet transplantation for treatment of the most severe cases of type 1 diabetes. He also worked to develop highly innovative strategies to transplant cells and organs without continuous requirement for anti-rejection drugs. In 2020, he led an international team that successfully completed the first FDA-approved control trial to treat the most severe cases of COVID-19 with stem cell infusions that were recently approved by the FDA to move to a phase 3 trial. He recently launched the Fit4 pandemic program to become resistant to severe viral infections, to halt progression of autoimmune diseases such as type 1 diabetes, and to prolong healthy lifespans. Dr. Ricordi founded the Diabetes Research Institute Federation, coordinating cure-focused research at institutions worldwide to eliminate geographical barriers to scientific collaborations. Dr. Ricordi has received numerous honors, including the 2001 World Prize in Surgery, the Outstanding Scientific Achievement Award by the American Diabetes Association. He was also knighted by the President of the Republic of Italy and inducted into the National Academy of Inventors. Without further ado, I will start the questions and again welcome both icons and pioneers in this world. And I hope I get this chance again, but I want to start off by saying thank you, because you have given us the hope for our kids that we have wanted and hoped for, and we're still looking for. So I thank you for that hope that you have given us. Here is my first question, and it goes to both of you. This is going to be really just us discussing things. Most of us, or many of us, probably have heard of the Edmonton Protocol, but I get the feeling that you both started long before that on your journey together. When did that start, and how did you start collaborating?
Okay, maybe I can kick off, and can I say first of all what a privilege and pleasure it is to be together this evening and with my dear friend Camillo. So Camillo and I began collaborating a few years before the Edmonton Protocol, because we could do all kinds of things in the lab, and he was doing all kinds of things in the lab in Miami. But there was one thing that we couldn't do for a while in Edmonton, which was to isolate good quality human islets. And part of the challenge was the quality of the enzyme that we had access to at that time. The collagenase enzyme was not very pure. And we really struggled working with Ray Rajotte and others to try to get islets to work. So I called Camillo up one day. I didn't know him before. And I said, would it be possible to come and watch some islet isolations in Miami and learn from you? And his immediate response without hesitation was, yes, absolutely. Come on down. So myself and my friend, colleague, Jonathan Lakey went together. He let us video record all aspects of the isolation steps. We learned every aspect from him in terms of the recording method. And as a result of that, we were able to isolate high quality islets in our own hands in Edmonton, because he was so willing to share so generously his protocols, his time, his team. And that really was the beginning for me of a really wonderful collaborative friendship that we've continued ever since.
Yeah, I may add that it's such an honor and a privilege to be tonight with James, a long-time friend and collaborator. As a matter of fact, before James entered the field and then became the absolute leader worldwide for number of transplant and success rate, we were having sporadic successes. We did the first successful trial in Pittsburgh in 1990, if you wish, put in the flag that you can reverse diabetes with an allograft. Minnesota already did auto transplantation from the same patient after chronic pancreatitis. But when we moved to type 1 diabetes, the problems started occurring, and that's when our collaboration became even stronger, because we share freely our methods, equipment, anything that we could contribute to the field. But has been really such a synergistic effort, because often, like up to now, I've been sharing things even before they're published with all the patent rights of everyone, never taking a penny for royalty. Beating even Banting and Best that sold their patent for $1. I did it for nothing, no dollars. But it's been like, thanks to James, that the field really moved forward, because he was able to demonstrate that with a modified immunosuppressive protocol and putting sufficient islets, like from 1 or 2 or 3 donors, you can always reliably achieve insulin independence. And this gives such a renewed enthusiasm for the opportunity to do eyelid transplantation. And then we had a very interactive, like James is one of the first that joined the Diabetes Research Institute Federation that we started many years ago, with the objective to promote international collaboration on the path to a cure and try to overcome the barriers to the development of cures. And it's been an example of a wonderful collaboration. I'm so happy that now even the foundation can finally collaborate and synergize effort to support our research and that of our collaborators. But I think has been a fantastic collaboration also, because in the United States, we don't have eyelid transplantation is not approved. We're still considered like drugs and not like an organ, and is not reimbursed as a procedure. So often we found ourselves, like at the time of Campath 1H came out and we started doing 2 or 3 transplants that work, and I called James and say, James is working with Campath, you should try again, don't give up. And then he does 100 patients with Campath the next year and publish and makes the next quantum leap. So it has been really terrific that we share ideas even at the beginning, and that whoever can realize it, we don't care who will get to a cure. We want just to get there in the fastest, most efficient way possible while collaborating, and then we'll both share and celebrate together when we cross the finish line.
So the Edmonton Protocol happens. People start to realize that everything that we're seeing today in many different areas, the idea of taking cells and putting them in other people and they're working, or recreating cells through stem cell lines. So we've gone through this whole history. Tell us, where are we today with all of this, and where do you both see us going to get to that elusive finish line? And I promise no one's going to ask you, when are we going to get there?
Yeah, good. Because that's the hardest question of all. I think what we've shown so very clearly with islet cell transplantation throughout all the years, from Camillo's beginnings, the early successes, and where we are today, is that cell therapy really can deliver a potential cure for this disease, for type 1, potentially for type 2, and for other forms of diabetes. It's a very simple concept. The cells in diabetes, patient requires insulin. That's because the body is not making enough, or is resistant to insulin and then not able to make enough. By simply replacing those cells with new ones, we can overcome that problem. The problem that we've faced as we move along with this is we use organ donors. Those are foreign to the body, and anti-rejection drugs are required to prevent rejection. And then because type 1 diabetes is an autoimmune process, the body can attack itself and potentially attack those cells once again. So I think we've learned so far that cell replacement therapy can be highly effective for patients, can provide incredible, stable, perfect or near perfect blood sugar control from moment to moment, day to day, but it's not always durable, it's not always long lasting, but it's been a very successful program. And we're trying to move now from a situation like that to being able to deliver this freely to many, many more patients, and with much less risk, so that the patients don't have to be on powerful anti-rejection drugs in the longer term.
Yeah, and I mean, very little to add. Like the next quantum leap. Now, eyelid transplantation is a reality. We've shown over and over again. We completed a phase 3 FDA trials of eyelid transplantation to treat the most severe form of diabetes, where you can justify immunosuppression, anti-rejection drugs. We have shown that the 20-year patient survival following an eyelid transplant is even superior to that of being on insulin without immunosuppression. So the risk of immunosuppression long-term is a relative risk. It depends when you start the immunosuppression. So you would never do it in a child or young adult. But if you're in your 40s or 50s, the likelihood to be around in 20 years is actually higher with an eyelid transplant despite immunosuppression. So that was a tremendous finding that both in Miami and in Edmonton we proven analyzing the long-term results. But now if you look, what do we really need as the next quantum leap to really make this biologic replacement available to all patients with not just type 1 diabetes, everyone requiring insulin, whether you had the removal of the pancreas, surgical pancreatitis, or type 2 diabetes that you start needing insulin in the long term. And these two big steps are first, developing an unlimited source of insulin-producing cells, such as those from stem cell-derived islets that you heard very recently also in press releases and big announcements, or in modified genetically engineered pig islets that can be rejected or have the same immunogenicity or less immunogenicity than human organs. But this could represent an unlimited source of insulin-producing cells to overcome the limitation of organ donation. Because if the second quantum leap is tolerance induction, so the moment we'll be able to do eyelid transplantation, whether from the pancreas of multi-organ donor or whether from stem cell-derived islets, and you'll be able to do it without anti-rejection drugs, everyone will want an eyelid transplant. And then will become like a lottery system, because today, like in the United States, we have over 11,000 people die every year for lack of suitable organs. And this is for immediately life-saving procedure. But think, if you now have only 1,000 pancreas available and millions and tens of millions of patients that will want an eyelid transplant. That's why these two pillars are so critically important now and we need to continue the interaction and the collaboration. So we can get more in the detail of some of this technology and strategies to achieve these two major pillars and the two major quantum leaps that we need to resolve before this will become, we'll be able to call it a cure.
So how, just bringing, and this is actually a question that came in right at the right time, is what's a more tangible way of describing how we solve that supply problem? What would actually happen?
Well, James, do you want to go first or I go?
No, go ahead.
So I will be presenting in Stockholm at the ESD, that is the largest Congress on diabetes worldwide, with over 23,000 delegates from all over the world, the results from the file for the initial Vertex trial with stem cell derived islets. And this has been already, I cannot share anything that is not public, but this has been an unprecedented results. And the comment when it was initially presented at the American Diabetes Association Congress in New Orleans was that these were the most important 15 minutes in the history of the American Diabetes Association. That gives me still chills when I repeat that comment. But it is really remarkable that you can finally, with a stem cell-derived islet transplant, reverse diabetes and obtain insulin independence. And it's still like, we're still away from saying this can be reproduced in hundreds of thousands of people and scale up to that level, but it's the proof that you can possibly have an unlimited source of insulin-producing cells. And besides Vertex, the ViaCyte trial have been also transplanting cells in many patients, and now hopefully even like we're collaborating between foundation and academic center, it looks like even Vertex and ViaCyte may be united more than ever instead of competing. So I think is a huge step in the right direction, because also it indicates for the first time that major companies are putting substantial funding that we would never dream to be able to collect through donations. So while donations are critically important to bring us to the level where these big giants get interest and put then the amount of volume of resources that is necessary to make it a reality for many patients, but still is critical because innovation comes from academic center, initial trials supported by foundations like the DRIF Canada and DRIF Miami, but then we cannot do it alone. And I think for the first time in history, the fact that you have major pharmaceutical company like Vertex, Eli Lilly, Novo Nordisk, Takeda, you have giants that are entering the field of biological replacement. You have Nobel Prize winners like Yamanaka, and so it's really a fascinating time because you can really see the power behind what we've been trying to develop for so many decades, and finally the light at the end of the tunnel of a cure. So that I will not say the cure is in the next 5 years, because I just watched Lisa Herpner human trial documentary that has been quite a remarkable venture, but also a sad story of participants to trial that may eventually not succeed. But it was a step in the right direction, and now I'm convinced that the next 5 years you will see things that you'll never expected before, given the history of failure, and in a way like resilience and perseverance in the cure-focused research. And resilience and perseverance is mainly for patients, because they and their family had to go through this roller coaster or hype, and then being sorry for lack of substantial progress. But then, the progress towards the cure has been going like this, but it's going definitely, the vector is going in this direction. And so I've never been so enthusiastic in my entire life. And I was just talking with Norma Kenyon, who also has a daughter with type 1 diabetes, and said, this is the first time I can really think we will be there in 5 years, but I'm not saying it loud. So
Dr. Shapiro?
Sometimes it only takes one patient to prove a difference. You see a patient who struggling with diabetes, next day after a cell transplant is leading a normal life, a life they never even dreamed of. And you see that happen over and over again. And now the possibility of being able to do the same thing, not dependent on organ donors, but with a limitless, in theory, a limitless supply of cells, this is possible now. It was just a pipe dream years ago. Now it's proven to happen, one patient after another. And now our job, I think, is to accelerate that progress even further, make sure it's safe, make sure it's even more effective as we treat more people.
And what do you both see as the expected progression of the trials? And will they start out as many trials just in adults with a certain criteria to prove that they're safe, and then build from there? Is that kind of how it will work, or do you see it going on a different trajectory?
I think most trials usually begin in adults because of the safety, efficacy balance and equipoise. So you don't want to cause harm. You certainly don't want to cause harm to a child who has an alternative, which might be injections of insulin. But as we move forward with these therapies and show that they're safe, show that they're much less risky than the previous therapies, then it becomes much easier to change that equation and include children in the trials. So I think we're always going to be cautious, but at the same time, move forward as fast as we can to include children when we can.
Dr. Ricordi, anything to add?
I concur.
Do we see the possibility? I know we saw this a little bit in the COVID trials, that the FDA can fast track if both of your work starts to prove productive and they start seeing it working. Can the FDA fast track this to get it done in bigger numbers and a faster result?
The FDA could certainly, we have to do a lot of work, both education and motivating. Like there are, so far FDA has been sort of blind towards considering islets like organ transplant, and we've been now still not giving up. In United States is the only country where eyelid transplantation is still considered like a drug, and we are considered like a drug company, so that you have to go through this biological license applications, spend tons of money, make the procedure, then hundreds of thousands of dollars for patients or insurances, and jeopardize even the possibility of reimbursement, if you wish. Well, in Europe, in Canada, from France to Italy, Switzerland, in Asia, is considered as an organ because islet, if you wish, they're comprised of 1,000 to 2,000 cells, are microorgans. They have their microvasculature. You don't do anastomosis physically, but they reconnect the vessel from the islets with the vessel from the recipient. And they comprise all kinds of cells, endocrine cells, pericytes, mesenchymal stem cells, endothelial cells, and this is really the smallest organ that we have in the human body, but in hundreds of thousands. So to try to fit eyelid transplantation in the shoes of a drug where you have standard manufacturing, raw material that are always exactly the same. Well, in eyelid transplantation, you have all donors are different age, different cause of death, different, is really killing at least centers and punishing patients in the United States, where now practically eyelid transplantation was halted because of this outdated FDA regulations. But luckily, like Canada can move forward and leave us in the dust. Europe, they already did randomized trial comparing eyelid transplant to insulin pump and artificial pancreas with incredible results in favor of the eyelid. And so the global collaboration that we're trying to set up is very useful in this direction, because you can be occasionally stopped in a country because of outdated regulation or political things like used to be for stem cell, but eventually you can continue elsewhere. And as far as we keep helping everyone who wants to get to a cure in the fastest, most efficient way possible to do so, that's our mission. We don't care if the cure will come from Miami, Edmonton, or Milan, or Lille, or London, or Asia. We just want to help anyone willing to be in the fight to get there as fast as possible.
Does it, you touched on something before, Dr. Ricordi, I just want to go back to, how much influence is it, or how much should it be, if Dr. Shapiro in Canada has success in a program and we want to mirror the same thing to help expand that in the United States? Does the FDA look at successes in other countries in their considerations to do things here in America?
Well, yeah, this is already, I don't know that FDA is looking, because they sort of saying we go our way or the highway, and it's not that you can show always, like Europe is going different direction, Japan is going different direction, Asia, Canada, and but what you can do is, like, you can expand trials and have sites in Canada, United States, and then if you're blocking United States, like we're considering, we have other trials ongoing where Canada FDA is allowing the transplant to be done, let's say, every 30 days, while here we're blocked and do every 90 days. And the patient inclusion-exclusion criteria, FDA still thinks to apply the inclusion-exclusion criteria we used for eyelid transplantation 20 years ago, imposing that you have to have a series of severe hypoglycemic episodes, hemoglobin A1C cannot be below a certain level, and say this is unrealistic in modern diabetes care, because now you have insulin pump connected to the glucose sensor, you have a low glucose suspend insulin infusion, you have alarm when you start getting hypoglycemia. So it's really difficult to find candidates based on the criteria of 20 years ago. And luckily so, because technology has been improving a lot. That doesn't mean that we still shouldn't remain focused to get to a final cure. And so that's a perfect example on how different FDA in different countries can apply different, can be a little more brain flexibility and a little more open to evolve more rapidly with aligning the regulatory guidelines. So I'm happy that we have this international breadth with the Diabetes Research Institute Federation and all our collaborators worldwide.
It's been a very frustrating journey watching the FDA and throughout their 20 years or so, consistently blocking our ability to move forward with islet transplantation. We've spent nearly $100 million in these phase 3 registration trials working together to demonstrate how safe islet transplantation is. We've treated literally hundreds upon hundreds of patients with an incredible safety record of this procedure. And yet still the FDA cannot grasp the concept that this is not a drug. It's a cell transplant treatment that is remarkably safe. And, I understand that a university manufacturing facility is not the same maybe as a large pharmaceutical company, but that's been a roadblock for us and has set things back and cost, I think, an enormous amount of research dollars that could have been spent perhaps better elsewhere. But trying to put that behind us, I think we've got an opportunity now as we free ourselves from the methods that Camillo developed for mass islet isolation from patients to the ability to make these now in the dish. Perhaps we can move on to a new journey with the FDA and with all of our international partners as we move forward with the trials.
Are you able to take, is it a pipe dream or a possibility to take someone's own cells, re-engineer them so they have insulin producing cells and put them back in the same person so the rejection ability is lowered greatly? Is that?
So that's the theory. And that's something we've been working on very intensively in my own lab over the last 3 years or so, making islets from patients' blood. When we first began that process, we had no idea how to do it. We didn't know whether to do it from skin biopsies or from which cells in the blood. And I think we've learned through a lot of collaborations and reading a lot of papers as well over the last 3 or 4 years, how we can do that fairly reliably and repeatedly so that we can now make patients' blood-derived islet cells that make insulin from patients with type 1, type 2 diabetes, and post-surgical diabetes. And at this point, we've been able to transplant those back into diabetic mice and reverse the diabetes in mice, but we haven't yet reached the point where we're ready to do the first in human trials, putting patients' own cells back in. Now, in theory, if it's a patient with type 2 diabetes, putting the patient's own cells back in should be very effective. If it's a patient with type 1 or autoimmune diabetes, it might work, it might not, and it might need some additional help with some minor tweaks to prevent the autoimmune process from coming back. And these are the experiments in the first patients as we move forward that we'll be able to answer that more effectively on.
Somebody just chimed in that they're happy to see that research is actually going to be impacting people in the type 2 community as well, because it's always been type 1 and we're very loud. Us parents, we're pretty loud about it. And so can you just walk us through some of these projects that are going on, that what possibly the type 2 patient might see and how it would work in their lives?
Well, I think for a patient with type 2, it'd be the same process as I was just described. Take a sample of blood, manufacture their cells, mass manufacture them to the point to provide a therapeutic dose for the patient, deploy that dose of cells in a place in the body that we think is most safe. It might be inside the liver, it might be under the skin, it might be in the other sites that Camillo's developed, like the omentum, and then watch those cells work without the need for anti-rejection drugs. It's a lot simpler in a patient with type 2 in some ways than it is in a patient with type 1.
So you have it's simpler in type 2 because you don't have autoimmunity. So the problem that you're raising, if you use your own cells and become beta cells and you have an autoimmune disease underlying, your body is primed to attack and destroy islets, so that you solve the problem of rejection, but not that of autoimmunity. And David Sutherland in Minnesota showed it with the transplantation between twins, that we say, well, between twins, you don't need anti-rejection drugs, and instead you lost the transplant within 2 weeks because of recurrence of autoimmunity, because your system is primed to attack your own cells. But you can possibly have cells that are not susceptible to autoimmune recurrence, and what James is working on, and we are working with Juan Bendala and team on generating new insulin-producing cells from the DAC precursors. And so there is a lot of going on regeneration of insulin-producing cells from the patient's own cells. In type 2 diabetes, you have the advantage that you don't have underlying autoimmunity, but you have the disadvantage that you have insulin resistance. So you cannot just continue to pour more and more insulin-producing cells, but you can combine strategies to address insulin resistance with a cellular therapy, biologic replacement. But as in many fields, including type 1, but in type 2, even more, there is no cure without prevention. So the moment you have a successful therapeutic strategy, you're dealing with preventing disease recurrence. So the same factor in type 2 that induced type 2 diabetes to begin with, and that in over 50% of the cases is preventable by just physical exercise, diet, proper nutrition, protective factors, like we're studying now for both type 1 and type 2 diabetes, is critically important, even more if you do an eyelid transplant in type 2 diabetes. And sometimes you see patients receive an eyelid transplant and they say, okay, now I can go out and eat like there is no tomorrow, French fries and ice cream. But those are the same lifestyle and nutrition and dietary pattern that bring 90% or more of Americans above age 65 to have chronic degenerative disease conditions that are not age related, but are preventable in most part, because our diet and lifestyle have evolved in a way that now the phase of decline, like for the first time in the United States for the last 3 years, lifespan has decreased instead of improving. For the first time, the children born today may live less than the generation of their parents. So these are alarm bells and red flags that should make us concentrate not just on the cure, but on how you prevent disease recurrence, how you address insulin resistance when we will do this transplant in type 2 diabetes. And we are going in the right direction also for this aspect, I believe.
Is this process applicable to other disease states? Do you see it applicable for, and if so, can you just give an example where you might see it?
Well, autoimmunity and autoimmune diseases like type 1 diabetes. Type 1 diabetes is the tip of the iceberg. Like we used to say, why do you concentrate only on type 1 diabetes? Is less than 5% of the 430 or 460 million worldwide with diabetes. But actually, type 1 diabetes is the tip of the iceberg of the autoimmune pandemic that affect 20% of the population with 100 different disease conditions. So it's what we are working for type 1 diabetes may very well be applicable to many of the other autoimmune disease conditions, and prevention of autoimmunity, halting progression of an autoimmune disease, or correcting and then preventing disease recurrence will be strategies that are likely to be very similar.
So the autoimmune process is always there. You have to then figure out a way to integrate.
You can correct it, you can reverse it, you can induce tolerance also and restore self-tolerance, like you can induce tolerance against transplanted tissue, but you can restore self-tolerance. And I hope we'll have time to discuss also some of the tolerance induction strategies that are key as the second pillar. Sorry, James, you were interrupted.
No, but I was just expanding on the point that the cell replacement therapy for diabetes is really just one example of regenerative medicine, and there are many disease states where replacing disease cells with new cells would offer an opportunity to cure those disease states too, and using the same kind of processes for cell manufacture, but perhaps not making islets, but making heart cells or making nerve cells or making other regenerative kind of cells. It's the new frontier in medicine that's coming.
So when you've been successful in animals or humans on blocking the autoimmune process, how long before it starts to slow down or stop and the body gets attacked all over again, or we're really in the process of trying to figure that out?
Well, I think that I see also a question from the audience. Does the autoimmune response wane over time, or is always there and is related? So like, I think that there are two strategies. You can wait after the acute phase when you may not have any more autoantibodies very active, so that will be easier to do biologic replacement without dealing with a very strong autoimmunity. But you can also think about strategies to restore self-tolerance and then eliminate an active autoimmune process, and then do biologic replacement to replace the insulin-producing cells that you no longer have. So his research goes hands in hand, like what is done by TrialNets or other groups to try to halt progression of or reverse autoimmunity at the onset of, even before the onset. Like now there was a study that showed that omega-3 fatty acid, for example, that have been helping also COVID and been associated with improved lifespan and decreasing mortality for all causes. But they've been shown that in patients that are antibody positive, in subject antibody positive, not yet diagnosed, but they're progressing towards diabetes, you can have a fourfold decrease of progression of towards autoimmunity. And that has been important because it shows how natural substances are re-equilibrating our nutrition, protective factors from the vitamin D, fish oil, our diet and other lifestyle component could really help us not only prevent autoimmunity, like the vital studies show that you can reduce 39% the incidence of all autoimmune diseases just with omega-3 and vitamin D, but you can do much better. And I'm sure in the next few years, you will hear much more in this direction.
How do you both see your collaboration moving forward? How the research that you'll do together, how actually do you see that, or is that sort of in process too as you work through the synergies between the two of you?
I think it's dynamic. It's ongoing. We strive for ways to work together, and I'm always excited to work with Camillo. I mean, one example would be the collaboration that he set up for us through Eyelid Pharmaceuticals to use a tolerance-inducing antibody in our eyelid transplant patients, a strategy that he and his team developed in Miami, but because of limitations, we're not able to take forward immediately in Miami. And he immediately shared that with us, and we've been able to get approval for it and found it hard so far to find patients to enroll. But there's one example of a dynamic collaboration, picking up the phone and just having that opportunity to work so closely together on such a regular basis.
And besides, Eyelid is a great example on how we work for over 20 years with Norma Kenyon, and I'm showing how this co-stimulatory blockers, so-called this antibody anti-CD40 ligand, could replace some of the most powerful immunosuppressive agents like Prograf. And then we have with eye tolerance that is now heavily involved, helping has been incredible. We had non-human primates off insulin and off immunosuppression of all kinds for several months. And we're moving towards clinical trial, hopefully in the next year or so. And this is the strategy is another collaboration that is combining the biohub strategy that we developed to recreate a mini pancreas in the abdominal cavity with the biologic scaffold on the omentum. And we combine this strategy with now a way to induce localized tolerance with this so-called fast ligand microgel. I don't want to get too technical, but it's a technology that was developed by Georgia Tech and University of Missouri in collaboration with Jim Markman at MGH. They did the first non-human primate transplant, and now we're moving towards clinical trials. So I think these are two entities to keep an eye, both Eyelid and eye tolerance, that is moving very fast towards clinical trial. And the idea is that you test initially with islets from multi-organ donor, but then if you can induce tolerance and remove immunosuppression, you will go straight to the real quantum leap that will be unlimited source of insulin-producing cells from stem cell sources. That's how there are two parallel tracks, but they will converge for sure in the next couple of years.
Okay, so I probably have time for just one more question, which might take a little time to answer. So I'll ask this, and it was a great question. Someone asked, what is around now that was not around 5 or 10 years ago? And why is now, at this point that you mentioned before, why is it that at this time you're most excited about this trajectory that's going on a different angle than it has? So maybe we can close with that one and see what both of your feelings, and then I'll ask you both for a final comment.
Well, maybe I'll kick off. What wasn't around 5 years ago were these embryonic and adult stem cells that I mean, they were around. ViaCyte had them 20 years ago, but they've been refined. Most recently in the last year or so, ViaCyte and CRISPR Therapeutics, have been carrying out the very first CRISPR gene-edited islet transplants using these stem cells. That wasn't around 5 years ago. That wasn't an opportunity that could be tested previously. And now we can dream and map out how we want our cells to behave, which genes we want active, which ones we want eliminated. we can go back to the drawing board with these companies, and it's a fairly efficient process to come back with these gene edits and then deploy them in patients and see what happens. It's an incredibly exciting time. This wasn't possible just a year or two ago.
I
Dr.
think that, yeah, I think first what we didn't have 5 years ago, as we mentioned before, we had the first in human demonstration that you can reverse diabetes and be off insulin with a stem cell derived eyelid transplant that was initially presented at ADA, and I will present in Stockholm in a couple of weeks, the 21st actually, then we'll be able to update you on that. But that is amazing to me because it's a quantum leap in the field, demonstrating you can do the same thing as an eyelid transplant from a multi-organ donor with a stem cell derived product. And the other very exciting thing is like tolerance induction progress, like from the eye tolerance with the fast ligand now having a month of non-human primates of immunosuppression and of insulin, to Bernard Herring with the strategy with the donor apoptotic leukocytes, also without immunosuppression for over 1 year. There are Eledon with the anti-CD40 ligand, and if you think maybe being able to combine some of the strategy to come up with a combination strategy that will be eventually the new gold standard is so exciting. This was never there 5 years ago, but what is unique is that everything we had in the pipeline, and maybe you heard about the same strategies in mice and in rodents. I remember when the eye tolerance strategy, when fast ligand was presented at the first JDF consortium meeting in mice and rat, I said, yes, sure, the number 440 way to cure a mouse that will not be translatable to human. But then you see the recent paper in non-human primates, say, oh, wait a minute, I never saw something like this in my entire career. And I called Jim Markman and I said, did you ever see something like this in your life? And he says, no, it's unprecedented. So this makes you excited that we're in a different stage. Everything is moving to clinical trial. We are no longer talking about small animal models or maybe pie in the sky, but also the other key factor to me is that major pharma industry bloc are pouring hundreds of millions of dollars in the field that would be unimaginable for us to be able to face. So we can come up with the invention, we can show proof of concept and principle in a pilot clinical trial. Maybe once in a lifetime, FDA funded a phase 3 trial, spending substantial amount of funding, and I don't think they will do it ever again. But the job is for us is to be innovative, come up with a new invention, proof of concept in a pilot clinical trial, and then industry will pick it up. And the big difference that was not here 5 years ago is that 5 years or 10 years ago, when I went to regenerative medicine advisory board for major big pharma group, they would say, why do you want to do it with a cell? What you can do with a molecule, you don't want to do with a cell, too complicated. And that now suddenly you have Vertex spending like close to $1.5 billion for this merging now, buying first SEM and now ViaCyte, and being able to put resources that can really make this trial go forward fast. And you have Eli Lilly is also investing a lot. Novo Nordisk has 200 people, I believe, in stem cell therapy in different areas, and you have many more entities, and we have, it's not just Vertex and ViaCyte. We're seeing all kind of new companies emerging from Europe, from United States, from Japan, and it's very exciting because it's finally the field is moving in the right direction and is moving translational. So never again to apply for a 5 years grant in mice and then reapply for another 5 years in mice, because that will give your children to go free to college because you get tenure in an academic institution. So we have to really change also the mindset. Basic science is critically important, but now is the time to be translational and go to the finish line through clinical trials and collaborating open access with anyone who can join the team.
Gentlemen, any final words? And then I'm going to ask Melanie to say a final word, and then I'll close. Anything before we close, Dr. Shapiro?
Again, as I said at the beginning, my dear friend Camillo, it's been an absolutely wonderful journey we've been on together. You shared so much with me. I've learned so much from you. We've also had an awful lot of fun along the way, fishing together and a few other things in and around Miami as we've tried to advance science, and working with Camillo to deliver on science has been so much fun, and it's going to continue to be so. And while there are serious aspects to what we do every day, it's been a real privilege to have fun with him and to advance the field at the same time. So a big thank you to you.
Well, thank you, James. It's been really tremendous. We've been leading by example, saying, what can you do through collaboration instead of being secretive and working? There is no advancement that can be done on yourself. I think that the biggest constant has been change, and change is progressing exponentially, and you cannot dream like the old academic institution will say, keep your chest to your chest of discovery, because that is your competitive advantage in the next round of competitive grants from NIH or JDF, and instead to be able to challenge this system while promoting collaboration, open access, exchange of everything we know even before it's published, and it has been tremendous. And I see comments in the chat on what about Doug Melton? This is exactly Doug Melton. We've been collaborating with Doug Melton and the Harvard Stem Cell Institute since 2014. Before was SEMA, now is Vertex, and now ViaCyte will join hopefully Vertex soon. And is an example on how the results that I will present in Stockholm come from Doug Melton research and the expanded trial, now multi-site, like the ViaCyte results have been presented, and the gene editing technology that will enable us to engineer cells and make them less immunogenic. There are so many exciting things, and as I mentioned before, the most exciting of all is that are all translational. We are not talking about 5 years of mouse experiment. We are talking getting into human. Eye tolerance, hopefully we'll have the first tolerance induction trial within a year, and Eyelid also, and Bernard Herring probably also with the apoptotic donor leukocytes, and we have to work all together. And most of all, the ability to continue to work with James Shapiro and the Edmonton team will continue to make breakthroughs and lead by example, because in our field, you lead, follow, or get out of the way. So something that I invented many years ago for the car industry, and now is applying to cell therapy too.
Melanie?
I'm going to answer the question, what didn't we have 5 years ago that we have now? And, we talk about COVID and pre-COVID, and we wouldn't have done this event. It wouldn't have been something we would have thought we could have done. And with bringing Zoom into the world and having the ability to bring together all of us in different parts of 2 countries to have this conversation. And, I'll take off my DRIFCAN executive director hat and I'll put on my mom of 2 kids who live with type 1 diabetes. This is hope. And so this is why I do this. This is why this is important. And I've heard Dr. Shapiro speak, and I get goosebumps every time it happens, because I know this is happening. And so I know how the diabetes community is feeling. And, back in the day, I've been doing this for 19 years with my kids, and we had hope, but there wasn't a lot of hope. We didn't know it. Just kept being told 10 years, 10 years, 10 years. And now finally we're seeing trials that are really making a difference, and we can move this forward. We really can. And Dr. Ricordi, thank you so much for taking the time to come together with us as well and have this conversation. It's very exciting to have you both on the same platform and hear this. And I'm also seeing the chat, and it's just constant. And that is the diabetes community who wants this successful. We can do this. We just have to do it together. And like what didn't used to happen in the past was diabetes organizations coming together and partnering and moving this forward. It's not about who does this and who gets the funding. It's about getting it to the right place so those people can find the cure. And we really do believe in both of you to do that. And so it just makes sense for us to work together to get this to the end. And so thank you to the DRI, to DRIF. I'm so excited that we're embarking on this, and to all the people who are on this call with us. Thank you. Thank you for your connections through our social media. Thank you for coming through and sending us comments. Thank you for reaching out to us. We need your help. Bottom line, people are asking, when is this going to happen? What is going to happen? We've got to fund it. So help us. And it doesn't necessarily mean, you know, you have to give a lot of money or any money. It's connecting us. It's just helping us get this to the final line with these two researchers. So thank you very much, Tom. I appreciate it. And I look forward to this communication, and this conversation will continue. We will try to look at something in 2023 as well. So thank you.
Well, I'm so overwhelmed. I really am. And again, as the dad cap 2 goes on my head, I thank you both for your work that you've done. You'll continue to do until we get there. Just a couple of house cleaning things to end everything. We certainly hope that you enjoyed this as much as we hope presenting it. If you have questions that did not get answers this evening, or you'd like more information, please go to either drifcan.com or diabetesresearch.org. Ask the questions in there. We will get it back to you in a follow-up email. We hope that this isn't the last time that we'll join for these educational programs. In fact, I'm being told now that we'll be planning one in the spring where Dr. Ricordi and Shapiro will provide an in-depth look at their respective research progress and projects that they're working on. None of this gets done without all of you who are watching. You're the funding sources that started these things. You're the ones that talked it out to the diabetes community. And for that, I say, on behalf of everyone else at our foundation, thank you, and please enjoy the rest of your evening.
Thank you.
Thank you very much.
Great to see you, James and Melanie, Tom, and everyone online.
Thank you. Bye-bye.
Thanks. Bye.